All right, in this video, we're going to discuss chapter 13. So we're going to discuss disease, different types of transmission, pathogenesis, and eventually we'll get into epidemiology. All right, to start off with, you're going to hear the terms infection and disease used interchangeably. However, they actually do have different definitions. So an infection by definition is when a contaminating organism invades the body. It also has to evade the external defenses and multiply in the host. So when we think about an infection, think about like a urinary tract infection. Usually the major culprit of a urinary tract infection is E. coli. What happens with E. coli is it ends up in an area where you would not expect it normally to grow. So thinking about that, that would be in the urinary tract. And because it now evades those body's defenses and now it's going to multiply in this area, that's when it leads to an infection. You'll also hear the term disease, and disease is going to be infections that lead to disease in this circumstance. And usually, whenever we discuss the idea of a disease, it's when normal body functions are altered, and so therefore, again, you'll see problems occur. So you can have kidney disease, or you can actually have other diseases come through from that original infection. Or you may hear of, for instance, like H. pylori, in the stomach causing gastritis or chronic gastritis, but it can lead to diseases of the GI tract. So, an infection is thinking about the organism evading the host defense, multiplying where it normally wouldn't be found. And because of that infection, it may lead to disease where normal body functions are altered. All right, now with disease, you're going to hear about signs and symptoms. Again, signs and symptoms are also used interchangeably, but a symptom is subjective, and so it's only felt by the patient. So if a person says that they feel fatigue, there's really no way to truly measure that. Same thing for pain, for if you hear nausea, if you hear numbness or tingling, there's no way to truly monitor that, and that's a subjective feeling on the patient versus signs are objective. So if someone has fever, vomiting, bleeding, high blood pressure, those are all signs. They're objective and they're measurable. And usually what will happen is signs and symptoms will run together in a disease, which we call a syndrome. Now, a person may also not have signs and symptoms, and they would be considered to be asymptomatic. So the infections are going to lack the symptoms. However, they may have some signs and they can still transmit from another host or go to another host. So just because they don't have those symptoms, they don't feel fatigue, they don't feel nauseated, they don't have the headache, that doesn't mean that they still aren't going to be able to transmit or pass on this potential pathogen. So again, just some terms that you'll use kind of interchangeably, but they do have their own discrete definition. All right, and then we're going to talk about the stages of infectious disease. So what you're going to see is there's 5 stages. The first stage is called the incubation period. This is when basically the person will come into contact with the infection, and this can last until they have their first signs and symptoms. So the incubation period all depends on the actual pathogen. Sometimes it may be 24 hours, sometimes it can be 6 months. It all depends on just the different microorganism that you came into contact with. But the time that you came into contact to when you have those first signs and symptoms is called incubation. Then what happens is you hit this prodromal period, and this is the first signs and symptoms, but usually this is going to be very general. So you may not feel great, but you don't have very apparent signs and symptoms of the flu or the cold or chlamydia, whatever that disease may be, and those signs and symptoms that would run with it. Prodromal is very general, like, I don't feel well, there's something wrong, but you're not full-fledged sick at this point. And usually the prodromal period is relatively very short, and it leads into the illness where now you have severe signs and symptoms. You really feel like crap. You have very apparent signs and symptoms, and they're very distinct to that particular infection. And then what will happen is the illness will run its course, and then eventually you'll start to have a decline. So your immune system starts kind of revving up the engines. You may be on a drug treatment at this point. Also thinking about microbial growth, they're losing sort of that area, those resources that they normally would have, and usually your signs and symptoms start to subside. And then eventually you'll hit the convalescent period, where there's no apparent signs and symptoms. They don't use the term cure, but convalescent is supposed to sort of display the idea that you're on the path of recovery and you don't have any apparent signs and symptoms. But in this time frame, you may ask, when is a person going to be infectious? And the truth is, they can be infectious at any one of these stages. You can be infectious in that incubation period. This is one of the reasons that STDs are so high, because a lot of times they have a pretty long incubation period and you don't have signs and symptoms. Again, when you don't really feel that great, the actual illness where you're truly sick, and then again in that decline or convalescent period. So you can be infectious at any of these stages, really depending on the pathogen. All right, so this just kind of gives you that idea of seeing sort of the timeline versus the number of microorganisms, or the intensity of the signs and symptoms. Again, an incubation period, that's the time that you've come into contact with the infectious agent to when you start now having signs and symptoms. Prodromal is going to be where you start to have, like you don't feel great, something is up, but you're not full blown sick. And then in the illness phase, again, you can see this intensity of signs and symptoms on an incline. This is where you feel the worst. And then eventually, whether it's the immune system, the treatments that you have, or again, just lack of space for those microbes to grow, you're going to start seeing that decline in signs and symptoms. And eventually, the idea would be that there are no signs and symptoms in the convalescent period, and that person is on the road to recovery. Now, talking about the road to recovery, we think about how to classify these infectious diseases. So you'll hear acute versus chronic. Acute, it's onset very quickly. So you immediately start to have signs and symptoms, start to not feel well, and usually it's short-lived. So this would be like the cold, for instance, or if you had like a 24-hour stomach flu. It's very quick. Again, you feel like crap for 24, 48 hours, and then it's relatively short-lived. And usually at this point, you are eradicating that infectious agent relatively quickly. But with chronic diseases, they usually develop relatively slowly. And because you're not really ridding yourself of the infectious agent right away, you'll start to see that it's continual, or it could potentially even be reoccurring as well. So the person can be sick for a longer period of time, but they don't have extreme signs and symptoms to boot. Usually, again, they don't feel great, but the signs and symptoms aren't 10 out of 10 horrible. Unfortunately, with chronic diseases, though, because you're not ridding yourself of that infectious agent, you're more likely to have tissue damage, such as what we would see with like chronic gastritis and H. pylori. You can see this with hepatitis or even with TB as well. It becomes more chronic, develops slower, or again, it can be reoccurrent, like what we'd see with like herpes simplex virus. Or I'm sorry, with hepatitis C. In latent stages, this would be that the pathogen is in your body and it can be there for a long period of time, but it remains inactive. And then, usually, due to improper diet or maybe issues with being immune compromised, then you can see that the virus itself will become active again, and now you have a reoccurring infection. So this would be the example of herpes simplex virus, or like for instance with shingles and that varicella zoster virus. So you can be fine for long periods of time, not have an outbreak for 6 months, and all of a sudden with herpes simplex virus, you can see that cold sore or those warts, or those genital ulcers reoccur. Same thing for shingles. So they call that a latent disease. All right, so now we have to think about, how do we acquire these organisms? And usually it's due to the idea of a reservoir of infection. So there's three different types of reservoir that we're going to talk about. Humans can be reservoirs, animals can be reservoirs, or inanimate objects such as like fomites, or even like the environment can be a reservoir as well. So a reservoir, by definition, is the site where pathogens are maintained as a source of the infection. And the reservoir may not be actually harmed by the pathogen. It could be just actively in that area and not causing them any harm. But if it gets transmitted to another, then you can see this infection take place. So like I said, there's three different types of reservoirs. Human reservoirs are very common. Again, we think about us actually maintaining these pathogens in the human body. And then people with signs and symptoms can transmit it. But also infected individuals that are asymptomatic, meaning again, they don't have those symptoms, can potentially be infected to others as well. Usually it's before or after the onset of illness that you're going to see. So a person that's asymptomatic may also still harbor that pathogen and be able to transmit it to someone else. And then you'll hear the term carriers. So carriers are the ones that are asymptomatic and they can be infected for years potentially. So this could be someone that potentially has HIV that's now moved into AIDS, someone that potentially has syphilis or even TB. Again, we'll hear about animal reservoirs as well. So usually when we discuss diseases that are becoming from animals and transmitted to a human host, they call it a zoonotic virus or a zoonotic disease. And there's about 150 of these diseases known at this moment in time. So this just naturally will spread from an animal to a human. And usually you have to acquire zoonosis through different routes. So usually you'll see it through animal waste, which would be the example of like E. coli, the 0157H7 particular strain. Again, if you think about animal bites like rabies transferring from one to another, if you were bit by a raccoon or a squirrel, and then also through insect bites. Now, when we talk about insects, we do call it an animal reservoir, but you'll also hear the term vector. Usually when we discuss bugs, in particular, or arthropods, we'll use the term vector. But if you think about it, for instance, if you were bit by a tick, the tick is actually going to harbor a pathogen that is a bacteria, and then you could potentially develop Lyme disease. It's not the actual tick that causes the infection, but it's the actual bacteria that they then are now going to lay into your body. And like I said, then there's these non-living reservoirs. So contaminated food, contaminated water, soil, those can all be reservoirs for infection. And usually the organisms are often due to contamination by feces or urine. And they can be occurring naturally in the environment. Like if you think about like Clostridium tetani or Clostridium botulism as well, those are normally occurring in the environment. And again, when we think about water as kind of a contamination source, usually it's again contaminated water or contaminated drinking water, like you could see with E. coli or Giardia. And then usually with like meats and vegetables, again, it's due to not properly washing them and you have contaminated food. So again, E. coli and salmonella. Salmonella is a huge one for poultry that you can see. And then when we think about these pathogens, most of them can't survive or actually remain active outside of their host. So a successful pathogen needs to overcome both defenses, what are called innate and adaptive immune defenses. And we're going to discuss that more in chapter 15. They have to overcome them long enough to actually multiply in the host. So not only do they have to find a portal of entry to get in, they also have to overcome our white blood cells in order to stay in that area, multiply, and then eventually exit the host. And it's usually a disadvantage for the pathogen to kill the host because obviously, if they kill the host, they killed their resource. And so it basically makes them lose their sources for nutrients. So again, it's not really an advantage for them to kill the host. Usually they want to keep them. So they would consider this to be this balanced pathogency. And then again, there are portals of exit. So these are a way to move to another host potentially. And these are sites through which pathogens can enter the body. So we'll hear about portals of entry and then we'll hear about portals of exit. When we think about entry, this is how they get into the host. And then portals of exit will be how they leave the host. So how they can get inside the host are usually through skin, again, like a broken or unintact skin, or maybe through a bite. We can also think of our mucosal membranes as well. So like the eyes, the mouth, inside the ears. We can think about placenta. So again, moving it from mother to baby. And then we'll hear about this paraenteral route as well, which usually is not considered a portal of entry, but it could potentially, normally it's not the typical portal of entry, but we'll make mention of it. All right, so again, when we think about skin, your skin is going to be made up of that stratified squamous epithelial tissue in that outermost layer, and that's supposed to be a barrier to most of our pathogens. But again, if you do have unintact skin, if you have cuts or potential like surgical punctures, if there was to be a burn, now you could potentially have pathogens get through those openings. And then others, like for instance, if we look at the scabies mites, they actually burrow into that layers of skin. And usually when you look at the mites or the scabies, they'll actually leave like little tracks, so they're kind of easy to identify. Like I said, we think about your the mucosal membrane, so we can think about the respiratory tract. We can think about the GI tract as well. And whenever you think of mucosal membranes, these are the areas that are lined in your body cavities. So again, eyes, mouth, ears, and again, even that GI tract as well. Again, we can think about placenta, so we can think about this crossing from mom to baby. And this is usually an effective barrier, usually, to make sure that pathogens aren't able to get in. But again, there are some pathogens that can cross that placenta, like Listeria for one. And this is one of the reasons that lunch meat should be avoided when moms know that they are expecting. They usually try to say avoid all lunch meats, and this is due to the temperature issues and cold cuts. And Listeria can actually lead to several problems like birth defects, premature birth, and even actually spontaneous abortion as well. Listeria is not the only one. There are several other ones that could cross over, but this is one in particular that we make mention of. And then you have that parenteral route. Again, this isn't typically a normal, like true portal of entry, but it can be bypassed. So this would be like if you had needles, thorns, nails, and what can happen is you're mechanically depositing the pathogen underneath those tissues. So again, like stepping on a needle or again, a thorn, something that again is sort of mechanically moving that pathogen underneath those tissues. All right, and then we have to make mention of those roads of transmission. So transmission is how, again, it basically moves from the reservoir or a portal of exit to another person's portal of entry. And so there's three major groups. You'll hear contact transmission, and I always think of when I think of contact with direct or indirect, usually this is if you are like directly touching someone or having direct contact with someone versus indirect, usually will have a fomite or like an intermediate in between. And then droplets would be like, for instance, if you were sneezing. With vehicle transmission, again, this is like the contaminated food and water that we think about, but it can also be airborne as well. And then, like I said before, when we hear the term vector, think arthropod. So think of transmitting via a bug. So either getting like a bite or a fly stepping in poop and then going and stepping on your sandwich. So contact transmission, we'll first make mention of direct contact. So if you are directly touching someone, you are in direct contact with them. So this could be the handshake. This could be through sharing of like a drink, kissing, sexual intercourse, oral fecal routes. All of those that there's no intermediate would be considered direct contact. Also, like if your kid sneezes on you, that's technically droplet, but we can think about that being a direct contact as well. Indirect is going to have like an intermediate in between. So this could be that someone sneezes on a doorknob or sneezes in their hand and then they touch the doorknob. So usually you'll see these inanimate objects. And inanimate objects we term fomites. So like if you had dirty bed linens, that would be a fomite. If you had a bloody stethoscope, or I'm sorry, like a bloody glove or a dirty stethoscope or like a dirty bedpan, those would all be fomites in particular. So chlorine doorknobs, glasses, utensils, those are going to create this indirect contact. And then finally, again, we make mention of droplet for contact transmission. So again, the sneezing taking place, sneezing, coughing, you're going to have the pathogen actually be transmitted through inhalation of these droplets. And so in this picture here, it's just kind of depicting the kind of how far away should you stand, and again, how close in proximity you should be. Usually you have to be in relatively pretty close proximity with those large infectious droplets. If you think about sneezing, it's about 1 to 3 feet. The smaller guys are going to be 3 to 5 feet. And actually these really tiny little infectious nuclei, you can be over 10 feet, and they can still potentially move to someone else. So like, for instance, cold and flu, you would have droplet contact transmission. All right, then again, we can use that vehicle. So thinking about airborne. Airbornes are relatively scary, so this would be like your measles or TB. And what can happen is you can have these coughing and sneezing droplets suspend in the air for long periods of time. And it's through this infectious droplet nuclei that this happens. So it's transmission of a pathogen that goes in the air and stays in the air, even after that person is long gone, and it can travel to another host. So again, measles, TB, usually we think about this as airborne transmission. And if you're in a hospital-based setting, they would actually do airborne precaution because this is really difficult to control. And then again, we can think about food or water being a transmission vehicle as well. So usually this will have an effect on the GI tract, and contamination can occur in a lot of different ways. Again, animals can harbor pathogens like Salmonella or E. coli. Unfortunately, it can also be added to food during prep. So if someone didn't properly wash their hands, you could have potential Staphylococcus aureus moving into those areas. You can have cross-contamination with meats and vegetables, or if someone was to have bad practices for butchering or handling of foods, and not having proper hand washing as well, you can also see some of these pathogens move into that GI tract. One really common thing that you'll hear about is foodborne intoxication. This is actually where you ingest an endotoxin. So it's the toxin that causes the illness. It's not the living microbe, because usually what will end up happening is the organism will become killed during proper like heating or grilling, heating it up to over like 160 degrees Fahrenheit. But some of these toxins are actually heat stable. So the ingestion of the endotoxin on growing food is what causes this foodborne intoxication. This is what we see with Clostridium botulinum and Staphylococcus aureus as well. All right, then it can be potentially a foodborne infection as well. So foodborne, again, you actually have to eat the living microbes, so you don't have properly cooked food. Undercooked chicken and hamburger are a huge one that we worry about. Again, it should be, I believe it's 160 degrees Fahrenheit for chicken and for hamburger, but it may potentially be higher. All different poultry and like beefs have different temperatures that are considered relatively safe. So that, again, you decrease that potential for foodborne infections. Signs and symptoms do not usually appear for at least a day, but then again, you'll start to have the diarrhea or the vomiting occur. And like, for instance, when we think about E. coli, that certain strain, that O157H7, usually thorough cooking before consumption will normally kill the organism. So again, it's just really, truly an issue of not cooking for long enough or eating too rare of meats, or just not having proper cooking techniques. All right, like I said, for water, this is usually going to involve the largest number of people. And in a big, huge fiasco in 1993, there was an issue with actually the municipal water. So this was in Milwaukee, Wisconsin, and it had to deal with this Cryptosporidium parvum that led to an intestinal parasite and really terrible watery diarrhea. And usually whenever we see diarrheal diseases, the issue of why they are so problematic and severe is because they can lead to hypovolemic shock. A person's not replacing their electrolytes, and it's really just the constant diarrhea and the electrolyte imbalance and water imbalance. With this Cryptosporidium as well, it has a relatively low infectious dose, which means that you don't have to have as many organisms to actually create an infection. In fact, this can take less than 10 organisms, which is what makes it so tricky as well. And again, although you have municipal water and you'd assume that it's relatively safe, about 400,000 people became infected. And then last but not least, we'll make mention of those vectors. So you could potentially have a biological vector or a mechanical vector. The two best examples I will give is malaria and then like a fly. Biological vectors are going to have it as part of their life cycle. So they're an organism that transmits and serves as a host. And so they have the microorganism part of their life. And usually this will involve a biting of someone to inject it inside that person's body. So if you think of malaria, malaria is due to a mosquito bite. The mosquito is not the problem. It's actually the protozoa plasmodium that is inside the mosquito. And when they are bit, they release that plasmodium into a person's bloodstream. And that person can develop malaria. And actually, it could potentially be deadly if they do not seek medical attention right away. Versus a mechanical vector doesn't have the pathogen as part of its life cycle. It's transmitting the disease through a mechanical host. So usually this is the thought of having a fly step in poop. Usually it's going to be poop. And then that can move into someone's salad or move on to like a ball or some other piece. And so when we think of chlamydia trachomatis, this is carried on the feet of a fly from an infected person's eye to a new host. And you can actually end up with this trachoma, which can lead to blindness. So there's sort of this just moving from point A to point B, but they don't have the pathogen as part of their life cycle. It's not inside of them. They're just transporting it from one place to another. All right, so when we think about now moving these pathogens out of a reservoir, this is where you hear portal of exit. Portal of entry, they have to get in. Portal of exit, they have to leave. And so many portals of exit are the same as the portals of entry. Pathogens often leave the host by body secretions or excretes, and human activities actually promote the transmission of organisms as well. So again, thinking about how these could potentially leave through tears, secretions, saliva, blood, mammary gland secretions, vaginal secretions, feces. These are all ways that potentially the microorganism can leave the host and now work its way into a new host via a portal of entry. All right, so that was going to be part 1 to this video. I'm making a part 2 as well, so stick tuned for that. Thank you so much and have a great day.